clinically relevant, everybody, right, is going to be dealing with HBA1C or glycated hemoglobin. And Tay Sachs, not everyone is going to be dealing with Tay Sachs because it's a very rare neurodegenerative disease. It's also genetic. All right, so you can read this for yourself, and we're moving on now to a review of glycosphingolipids, which we just looked at for ABO blood type, right? We just looked at these. And now we're back looking at these again in the context of ganglioside GM2. Ganglioside GM2 is a glycosphingolipid which is particular to the membranes of nerve cells. And you know, the cell membrane is constantly being synthesized, and those lipids are going to be broken down and renewed, right? So we have this constant recycling within the cell. Ganglioside GM2, in fact, your glycolipids, right, whether they are regular, triglyceride based, right, or whether they are sphingolipid based, they are being synthesized in the smooth endoplasmic reticulum, right? So go back and review endomembrane system of the cell. So these lipids become glycosylated within the endoplasmic reticulum. And then they're going to be, right, essentially gently exocytosed to the surface of the cell, where they will become, right, these lipids will become part of the cell membrane. So what happens when we're going to be breaking down these lipids? Well, the reverse is going to occur. So we're going to have essentially an endocytosis event at the cell surface, and that endocytic vesicle is going to fuse with a lysosome, right? And so these things are born in the smooth endoplasmic reticulum, I'll put synthesis, and in the lysosome is where they are degraded. And of course, we have enzymes that will be degrading the carbohydrate portion of the molecule, and other enzymes which will degrade the lipid portion of the molecule. So if we have this complex oligosaccharide on the surface of ganglioside GM2, we need specific enzymes to degrade this. And these specific enzymes have to recognize recognize, right, these specific sugars. Okay? So remember, carbohydrates is informational molecules. And so if we have different, sugar monomer units joined together with different glycosidic linkage types, we are making a very unique type of oligosaccharide, and we will need enzymes to degrade it, that oligosaccharide, which are specific and recognize that oligosaccharide. Okay, so let's take a look at this degradation pathway. And this degradation pathway that you're looking at here is focusing on degradation of the carbohydrate. Your ATE. So we're focusing on degradation of the carbohydrate. Again, this degradation is happening in lysosomes because this is where the enzymes for degradation, wow, I really can't spell today, enzymes for degradation are. And so this collection of disorders falls under the label, the general label of lysosomal storage disorders. And there's some great pictures coming up. Now, when it comes to things like, I should say, disorders like Tay Sachs disease, right, or Gaucher disease, or Sandhoff, or Fabry, or Niemann-Pick, well, I'm going to take Niemann-Pick off the table. When it comes to the ones that I've given X's to, these are defects in breaking down the carbohydrate head group. They are not defects in breaking down the lipids. Niemann-Pick, right, we're looking at, this is essentially a cleavage of, right, a lipid, right? So the enzyme that's defective there is sphingomyelinase, and so that's the odd one out. Everybody else that has the X, we're looking at a defect in an enzyme that is breaking down a specific oligosaccharide. As far as pathogenesis is concerned, and disease progression, symptomology, if you have a defect in an enzyme that's early in the pathway, the disease progression is worse. And so earlier defective enzyme is worse. Right? So if you have a problem with an enzyme early in the pathway, again, we're going to focus on hexosaminidase A here for Tay Sachs disease. That's going to be a more, how should I say, a worse progression than a defect with an enzyme later in the pathway. So let's move on and look at the next slide here. So let's take a close-up view of this hexosaminidase A enzyme. And hexosaminidase A is the specific enzyme that is defective in Tay Sachs. So this is the enzyme that's bad in Tay Sachs. And technically, this enzyme is cleaving away an N-acetylgalactosamine, right? So this guy here is N-acetylgalactosamine, and cleaving that away from this guy here is galactose. And so if you can't do that, you're going to have a buildup of ganglioside GM2 in the lysosomes. And so the lysosomes are going to get packed with this lipid, and that's going to eventually damage the cellular function. The cell will die. These lipids are pronounced on neurons. And so if we disrupt the functioning of neurons, we disrupt the communication in the nervous system. And so that's not good, especially if we're looking at things like cognition, right? If we're looking at regulation of bodily systems and functions. So this is not a good disorder. This is a disorder that is progressive, and we would say it's neurodegenerative. There is no cure. In the desire to make this a more familiar type of presentation to you, I went online and I started to look for personal stories of Tay Sachs disorder, and it was way too intense, I think. This disorder, it affects primarily babies, and these children very rarely live past age 4, and they lose the ability to see, to hear, to think, to interact, and they have seizures. It's a really horrible disorder, and again, there's no cure. There is, right, a genetic component to this, and because there is a genetic component, there are certain, world populations where this disease is more prevalent, and it's more prevalent in Jewish populations of Ashkenazi descent. The inheritance pattern is autosomal recessive, so you need to have 2 unhealthy copies of the gene, and there is prenatal genetic testing available for this. So that's just a heads up about the genetics. If we look at this gene, there's actually 2 loci in the genome that are required to make a functional hex A. So there is a gene for an alpha subunit, and that is on chromosome 15. And there's a gene for a beta subunit, and that is on chromosome 5. Now, hexosaminidase A requires 1 alpha and 1 beta subunit. So these are 2 protein chains synthesized from 2 different genes at 2 different loci in the genome. And, we can have hexosaminidase genes combine, right? The proteins combine to give us a different hexosaminidases with different oligosaccharide specificities. So hexosaminidase B consists of 2 beta subunits, and that can cut a lot of oligosaccharides, but unfortunately cannot release this terminal N-acetylgalactosamine from GM2. And then we have 2 alpha subunits that can combine to give us this hexosaminidase S. And unfortunately, hexosaminidase S is pretty useless. It's an unstable enzyme, and it really has very limited substrate specificity. So it's not going to cut GM2, and it doesn't cleave away, that N-acetylgalactosamine from other oligosaccharides either. So we really need to have 2 functional genes to get this hex A to work. If you look at this image here, and there's another picture coming up, this is showing you, this is a scanning, is it scanning or is it transmission? It's transmission. Transmission electron micrograph of lysosomes. And so this is a lysosome. This is a lysosome. This is a lysosome. And in those lysosomes, we see what's called these onion skin whorled membranes. And so we have the buildup of membrane that's been taken from the cell membrane. And this is because we are lacking hexosaminidase A within these lysosomes. We can't break down ganglioside GM2, and those components of membrane that contain large quantities of ganglioside GM2 are building up, and they start to circle back around upon themselves like onion skins. So that's that onion whorled lysosomal membrane appearance. If you want to look at, hey, this is ganglioside GM2, and you want to look at, a more detailed structural view of this oligosaccharide and where your hexosaminidase A would cut, you could see that here. And so this would be the bond that's being broken by hexosaminidase A. And here is your, we'll say, preserve the same. The N-acetylgalactosamine cleaved away from the core polysaccharide. This is that beautiful picture that I found on the internet. And here you are looking at the cellular morphology in different lysosomal storage disorders. And again, lysosomal storage disorders because these products are accumulating abnormally within the lysosomes. And so Niemann-Pick disorder, you get this foam cell type of appearance. And so you can see that this is looks like a beautiful puffy foam in this area. So we call that foam cell appearance. Again, here's your Tay Sachs disorder. Here's Niemann-Pick disorder, and you can see they're calling these things zebra bodies. And so you're saying, why is it a zebra body? Because there's stripes. See the stripes in here? It's hard to, right? See there's stripes in here. Try to find another one. See how there's stripes in here. So instead of having this onion whorled appearance, we have kind of sheets of lipids in the Niemann-Pick disorder. And anyway, Gaucher cells, crumpled tissue paper appearance. And so you could see within the cell in here, we have this crumpled tissue paper appearance. Anyway, it's interesting. That's it for Tay Sachs. Questions on Tay Sachs before we move on to HbA1c? So kind of the point for Tay Sachs was the answer, right, or the opposite concept to ABO blood type. In ABO blood type, we looked at specific enzymes that build these informational oligosaccharides. And in Tay Sachs, we looked at specific enzymes that degrade these specific informational oligosaccharides. So it's kind of two sides of the same coin. Okay, let's look at HbA1c, and everybody's going to be dealing with HbA1c because there's so much diabetes in the population. Okay, so HbA1c is monoglycated hemoglobin, and it's also referred to as GHB. Glycated I'll just put hemoglobin, right, for HB. So glycated. Glycated is an important word, right? Glycated. Non-enzymic addition of sugar. Non-enzymic addition of sugar, usually to a protein, right? So glycated hemoglobin means that we had just plain old regular chemical addition of a sugar to the hemoglobin. There was no enzyme that was needed for this to happen. So, you know, it's really kind of cool. Hemoglobin is a natural sugar sensor. Let's put it that way, in the body. If we look at red blood cells, and again, anytime that somebody says to you, well, where's hemoglobin? Never say it's in the blood, because that would be bad. Hemoglobin is inside red blood cells, which, the red blood cells are in the blood, right? Hemoglobin should never be free in blood. So in a red blood cell, right, red blood cells are just membrane-bound sacs of hemoglobin that float around in your blood. We have hemoglobin proteins, so I'm trying to make, right, alpha-beta hemoglobin. That's as good as I can do. And red blood cells are freely permeable to glucose. And so glucose can enter and leave these red blood cells. Are there glucose transporters? Yes, there are. And they are, pretty forgiving as far as allowing the passage of glucose. Glucose can come and go as it pleases in and out of red blood cells. So basically, the glucose level in plasma equals put the glucose level in red blood cells. And so HbA1c levels allow to measure, let's say, the control of blood glucose over time. So we'll say several weeks' time. All right, so if you're dealing with someone who's a diabetic and you want to get a good idea of how well they're controlling their sugar levels, I shouldn't say that. I should say how well they're controlling their glucose levels over time. HbA1c is a monitoring test that you can order. Normally, in most individuals, HbA1c levels are about 5%, meaning that if you look at 100 hemoglobin molecules, about 5% will have a glucose attached to them, so glycated hemoglobin. People who have well-controlled diabetes have a slightly elevated HbA1c level, and so they have about 7%. people with uncontrolled or poorly controlled diabetes have HbA1c at 13%. So 13% of their hemoglobin population will be monoglycated, have one glucose attached to the protein. Now, of course, that 13% number it can go up, right? But anyway, so where is the glucose attaching, right? Or, right, let's put it this way, at primary amines. And if we look at proteins, where are we finding primary amines? We're finding them, for example, on lysine residues. And so the most common attachment point here on hemoglobin for a sugar molecule would be at a lysine, right? We can also have attachments at the N-terminus of protein chains as well, right? So those are the places that you're going to be finding the sugar residue has attached. Now, HbA1c was molecule of the month for PDB, right? The protein data bank. And if you click this, you can go and read up on those resources there at PDB. If you take a look at this hemoglobin molecule, don't forget, right, your structure of hemoglobin. So it's, right, alpha 2, beta 2. This would be a heme in red. This is another heme in red. And they're showing you here in green, right, that's your sugar, right, the glycated hemoglobin, which is very commonly found on lysine 99. Now, why do we care? You know, glucose doesn't only attach to hemoglobin. Glucose attaches to other proteins too. And so we can have glycation reactions with those other proteins as well. So where it says protein, this is any protein, not just hemoglobin. And what happens is that there's intramolecular rearrangements. Are you responsible for all of this? No. I'm not going to hold you responsible for the mechanism here, but again, know that it's right. It's a primary amine, and here they're showing you with lysine, but it can occur with any primary amine. Can do this, and of course, here's your what you guys should know this, right? Glucose. There's your glucose, right? I'm D-glucose, and just about everybody on the Nearpods was getting that. So good for you. Now, the deal is that once we undergo this glycation and intramolecular rearrangement, we form these advanced glycation end products, right? So this thing here. These advanced glycation end products have their own receptors on cell surfaces. And they happen to be receptors on things like working cells of the immune system, so on neutrophils, on macrophages. And we would call those RAGE, right? So advanced glycation end product receptor. And people call those receptors, they call them RAGEs, because it just flows better, right? The RAGE receptors. And they can kick off these signaling pathways in the cells, so they're going to cause the production of cytokines, which are going to lead to all sorts of things like inflammation. And if we look at inflammation, you know, inflammation can be responsible for a lot of the collateral damage that's seen in diabetes. So kidney damage, damage to the retinas. Also in diabetes, there's a high incidence of atherosclerosis. And atherosclerosis, we have deposition of cholesterol in the walls of the arteries. That's another process that is relying on inflammation as part of the process. And so it's a really neat story about pathogenesis. So I encourage you to click this and watch the video. We're not going to do it now in the interest of time, but watch the video about the link, right, between these glycosylated end products, these AGEs, advanced glycation end products. Oh, I said glycosylated. That was bad. I should say glycated, right? Because it's non-enzymic formation here of these advanced glycation end products. So these are, right, glycated proteins. And once these bind to a RAGE, right, we can have that cytokine response, inflammation, which promotes cellular damage, and right, it's not a good thing. So that's the deal with HBA1C, and that's it for the medical applications here of carbohydrates. Questions? Medical applications of carbohydrates? Anything? Okay, so question from Preston. So to be sure, this is an intramolecular reaction that reacts with the nitrogen within the protein the glucose is attached to. This portion here that I'm circling here, all this is intramolecular. And again, this has to do with rearrangements that are centered on that nitrogen. And we're going to move through this structure here is your Schiff base. Again, don't worry about it. But once we have that interconversion, we end up with the formation of more of like a ketose, or I should say a ketose versus an aldose that we started with. So everything that's in this box, this is an intramolecular rearrangement here. When we start out, though, we have an intermolecular addition. And this would be between, again, it's a primary amine plus an aldose. Okay, so that's the differentiation there. Okay, and so again, once we undergo this rearrangement, these guys are very stable. Okay, and so that's the idea. All right, let's look at the homework, shall we? Look at the bioenergetics homework just so that you guys have an idea of, here I have to pull it over to the tablet. It's going to let me. There it goes. Oh, it won't let me pull it over. Okay, here we go. So if you guys want to get started on this, you can get started on this. And this is also leading us into this concept of bioenergetics and metabolism. So just remember, there is a there's a chemical basis or a molecular biochemical basis for every biological action. And so if we look at, questions 1 and 2 kind of go together. And so for questions 1 and 2, we're looking at this condensation of glutamate and ammonia to yield glutamine and water. So here you can see there's your glutamate. Where has the ammonia gone? Here's the ammonia, and we get glutamine, right? So if we're considering those different structures, right, I want you to depict the reaction in its thermodynamically favorable direction. So to go from glutamate to glutamine, we have a delta G prime naught of positive 14.23 kilojoules per mole. I think you guys are pretty good with the concept now that if we have a positive delta G, that's thermodynamically unfavorable. If we have a negative delta G, that's thermodynamically favorable. If we have in this direction that a reaction runs, we have a positive delta G. If we run the reaction in the opposite direction, we will have a delta G of same magnitude, opposite sign. So basically, if we reverse a reaction, we change sign on delta G, but keep same magnitude. Okay, so that's the deal. Right, I want you to depict this reaction in the thermodynamically favorable direction. So that's question one. Question two builds on question one. So we know that formation of glutamine from glutamate is thermodynamically unfavorable. But if we couple a thermodynamically unfavorable reaction to a thermodynamically favorable reaction, we could possibly make this overall combined reaction thermodynamically favorable, meaning that we're going to end up with a negative delta G. So this is going to want to then proceed from products to reactants. And, you know, delta G values for reactions are additive in sign and magnitude. Okay, so for example, if we said that glutamate plus ammonia is going to give us glutamine plus water. This delta G prime naught, right, and I'll explain that when we get to the lecture very quickly, is a positive 14.23 kilojoules per mole. All right. Now, if we add in here the hydrolysis of ATP, right, to ADP plus PI, that has a delta G prime naught. Ugh, let me erase some of this. It's terrible. Our delta G there is negative 30.5 kilojoules per mole. Now, if we combine these equations, right, we would have this, and usually this is written so what is the overall delta G prime naught for this combined reaction? Well, you'd have to do the math. Right, so if we have 14.23 kilojoules per mole And then we have negative 30.5 kilojoules per mole. You do the math, and you're going to end up with the overall delta G prime naught for the combined reaction. Got it? Yes, Preston has got it. Don't forget your units, right? Always make sure that you have the units. So always make sure that you're providing the proper units. And please always work in joules or kilojoules. You could also work in calories, but it's, more standard to just use joules. By the way, if any of you have Amazon Prime and you're looking for a great thing to watch, there's a short series. It's a 3-episode series called Precision, and it's about the history of measurement, and it's fascinating, and they talk about all of these units in there. So I've watched it a couple times. My daughter's getting upset with me. She said I can't watch it anymore. But anyway, Precision, right? The history of measurement. Questions on delta G before we go on. So basically, your key takeaway points here: delta G values for reactions are additive in sign and magnitude. And by coupling together a thermodynamically favorable reaction with a thermodynamically unfavorable reaction, we can, promote these reactions to go, in the direction we want them to. Let's take a look at the next question here so you have an idea, and then maybe we could at least get through the overview of thermodynamics, which is really stuff that you've seen in Gen Bio. All right, let's deal with this idea of, this is a series of 1-carbon molecules in different states of reduction or different states of oxidation. Depends on your viewpoint. Let's start with methane, which I'll tell you right now is the most highly reduced. And of course, we're pushing over towards carbon dioxide, which is the most highly oxidized. All right. Now, that's a covalent bond. And we say that carbon-hydrogen bonds are non-polar covalent. But, in reality, carbon is a little bit more electronegative than hydrogen. And so we don't like to think about that, but it's true. And so there are 2 electrons in this bond, right? And we say it's nonpolar covalent, but carbon is more electronegative. And so in reality, carbon is going to be a little bit of an electron hog. And so this carbon is going to hold onto those electrons more of the time. Same deal with all of, let me try to make it, all of those electrons. So carbon is going to hold on to both of those electrons in each of those bonds more of the time. Okay, so in methane, let's say that carbon has 8 electrons. Okay, now let's move over to carbon dioxide. In this case, oxygen is far more electronegative than carbon. And so this poor little carbon here is not going to have any electrons because oxygen is going to hold both of the electrons in both of those bonds. So oxygen here, plus its own valence shell electrons, is going to have all the electrons. And then up here, same deal. So the electrons in this covalent bond are owned by the oxygen, and the electrons in this covalent bond. Where did my... Hang on. Do I get a laser pointer here? No, don't. Okay, well, you get the idea. Right, so it's this bond, these electrons, this bond, these electrons. And so this carbon here, no electrons, right? Carbon has no electrons. So it's the most highly oxidized. Remember, oxidation is loss. Oxidation is loss. This carbon has lost all of its electrons. It is oxidized. It has no more electrons to give. So if you follow this logic, you should be able to put these other molecules, methanol, formaldehyde, formic acid, into a series from most highly oxidized to most highly reduced. So you're going to start with carbon dioxide. This is number 1 in your series. You're going to end with methane. That's number 5 in your series. And you're going to order all these compounds. So methane is the most highly reduced, right? Reduction is gain. Reduction, right, means that you have a lot of electrons. So this carbon is the most highly reduced because it has the most electrons. Questions on that concept? Okay. And then you're going to follow the table right here, and you're going to write in the names of the molecules here. You're looking at the free energy that can be released upon complete oxidation of the molecule. So obviously, right, if we have more electrons to lose, we have more energy to liberate. All right, and then for question number 4, you're using that same concept that we looked at. I think, do I have the numbering wrong on the questions? No, I don't. I'm good. For question number 4, you're using the same concepts as presented in question number 3. And for question 5, I'm going to give you the reaction, the equation that you need. You need delta G equals delta G prime naught plus RT ln products over reactants. If you want to get started on it, you can. Right? And you're looking at the same concepts, as we looked at earlier for what is like delta G prime naught. So I think you have enough to at least get started on the homework. Those of you who like to get a head start on it, you could start on that. How are we? Oh, it's 11:50. 1 minute. Let's take 1 minute and just look at this. Bioenergetics and metabolism, an overview and compartmentalization. So it's all about energy. Here's the learning objectives. You can read them for yourself. In the big picture, in the grand scheme of things, if we look at life, we really need the sun to have any life on this planet. It's all about, living things are classified according to where they get their energy, how they use their energy, and where they get their carbon atoms. So as far as this is concerned, you're here. We are in the same category, right, with a lot of fungi, protists, and bacteria. Basically, human beings are obligate aerobes. So we are obligate aerobic organisms. We do have some cells in our body that they don't care if they see oxygen at all, like our red blood cells. They're obligate fermenters. They don't care about oxygen, right? So we'll look into that too. But this is a review from Gen Bio. So again, we classify living things primarily by where you get your energy and where you get your carbon. And so if we look at, first of all, where do you get your energy from? If you get it from the sun, from sunlight, you're a phototroph, right? You're a light eater. If you get your energy not from the sun, that's the easiest way to put it, then you are a chemical eater. Now, let's just focus on over here, right? Because this is what we're dealing with. Where do you get your carbon atoms from? If you get them from air, right? From CO2, the gas, then you're a chemoautotroph. You're a chemical eater, and you pull your carbons out of thin air. And so in that case, we have, examples in the microbial world. Right? So things that you're not really going to deal with. There are some toxins that these bacteria produce that you might deal with syndromes from the toxins, but basically, not of interest. And then we have where we are. If you get your carbon atoms from organic compounds, you're a chemoheterotroph. So you are a chemical other eater. You need chemicals, right? Organic chemicals for your fuel source, and you get your carbon atoms from breaking down those organic chemicals. So you're eating organic compounds made by other organisms. Okay, and then we look at how do you use your energy. If you run an electron transport chain and you have oxygen as your final electron acceptor, then you're us, right? Obligate aerobic organism. If you run your electron transport chain, hang on. If you run your electron transport chain and you use not oxygen as your terminal electron acceptor, then you're an anaerobic organism. If you use an organic compound, right, as your terminal electron acceptor, you're a fermenter. And there are some subcategories here. So at least we got to introduce the topic. We're a little bit over time. Any final questions? I'm confident we're going to finish up with ATP on Friday, and we're going to be right back on track where we need to be with the lecture material. Anything. Okay, so for lab tomorrow, I will be online and available to you guys at 9:30 and at 3:30, right? So you have an online exploration of carbohydrate structure.